Good afternoon, everyone. Heartily welcome to all of you to the Caritas Pharma Talk 4. Caritas Educational Institution started its journey to pursue new opportunities in healthcare with an aim to widen the prospects of learning through innovative pursuits and constant endeavors. With a strong leadership and presence in respective fields, we strive to enable the young budding professionals to become globally competitive in terms of education and proficiency. Caritas College of Pharmacy started with an intention to create professionally qualified, technically competent, spiritually mature, psychologically integrated, and socially acceptable pharmacists with professional ethics, good human values, and national organs. So far, we have conducted 3 Caritas Pharma talks in the form of national webinars, which opened up an awareness session for both pharmacy teachers and students all over the country to renovate their ideas as well as to rethink and polish their brains. And today we have Dr. Benny Abraham Kaipare, who is an associate professor, Baylor College of Medicine, Houston, USA, for the Caritas Pharma Talk 4 on topic Mitochondrial Metabolic Reprogramming in Cancer. We are indeed greatly honored to have you here for opening a novel topic for discussion. Let's begin the formal session by invoking the blessings of Lord Almighty. Now, I would like to invite Dr. Sajid Jose, Academic Director, Caritas College of Pharmacy, to introduce the speaker and welcome the gathering.
Mr. Dr. N.A. Equity, Administrator, Dr. Sinu Thomas A., Vice Principal, respected teachers and students of Caritas, all respected teachers and students participating online from all over the country. Good afternoon to all of you. I'm very proud to stand in the stage of Caritas Pharma Talk, episode 4. The overwhelming support and participation received to Caritas Pharma Talks from all over the country in the previous episodes of made us to proceed further with the series of talks. I'm very happy to introduce the eminent scientist, Dr. Benny Abraham Kaipare. He is one who achieved golden heights from an average background of Kerala. He did his B.Pharm from St. John's College of Pharmacy, Bangalore, M.Pharm from JSS College of Pharmacy, Mysore. His PhD was from JIPMER, Pondicherry. After PhD, he worked a few years as assistant professor in Department of Pharmaceutical Sciences, Mahatma Gandhi University, Kottayam. Then he did his first postdoctoral fellowship at Dr. Margaret Fisher Bosch Institute of Clinical Pharmacology, Germany. Then he moved to Baylor College of Medicine, Texas, USA for his second PDF. Currently, he is associate professor in Department of Molecular and Human Genetics in Baylor College of Medicine, Houston, USA. His area of research interest includes mitochondrial crosstalk in cancer, mitochondrial energy reprogramming in cancer, nanomaterial for bioimaging, etc. He published more than 55 research articles in high-impact international journals, and his corpus H index is quite high, that is 28, with an approximate 6,000 citations. He is a role model to all of us. His determination and smart and hard work is the secret of his success. With proud and happiness, I wholeheartedly welcome my dear friend and colleague, Dr. Benny K. Abraham. to the Caritas Pharma for delivering today's topic. And I also welcome all the staff and students and those who are participating online to this meeting. Thank you all.
We are moving on to the formal session. Also, I would like to remind you, for those who are attending the session online, you will have the opportunity to submit text questions to today's speaker by typing your questions into the question pane or chat box. You may send in your questions at any time during the presentation. We will collect them and address during Q&A session at the end of today's presentation. And for those who are attending the session in live, if you have any queries, you can come forward with those at the Q&A session. We are privileged to have our speaker, Dr. Benny Abraham Kalpare today. Let's hear from him his views on this subject.
So what I was trying to say that whenever I come to pharmacy college, it's always an excitement because you have a multi-speciality, because you learn science, you learn chemistry, you learn pharmacology, and you are the future to make something exciting tomorrow. So today I'm talking about a topic which is about mitochondria. I try to, because I know that you are of different level of education in because there are junior students, senior students. So I'm trying to make it as general subject as possible to avoid more complicated signaling or very narrow molecular level findings. So I'm just going more with the phenotypic findings so that I can involve most of you in the talk. But still, if you have any concern or question or something you're not understanding, you're always welcome to ask me. Okay. So I'm talking about mitochondrial metabolic reprogramming in cancer. So why mitochondria? So you will understand why mitochondria is so important in cancer during my talk, I hope at least. So where I'm coming from Houston, and Houston has such a wonderful place that's called Texas Medical Center. The advantage is that there is one of the world's largest medical center where all the institutions are almost in one campus. So that's I think the world's one of the biggest medical centers. For example, here you can see there is Baylor College of Medicine here. Where is my pointer? Can you see my pointer? Can you see? Okay. So this is Baylor College of Medicine, and we have MD Anderson Cancer Center. We are just across the road. And Texas Children's, St. Luke's, Methodist Hospital, Rice University. All our big institutions are the same campus. So it's a very beautiful environment because you always have very high talks going on in one of the other institutions. There are a lot of faculties around to talk with, and it's always a pleasure. So, so that environment is very good for doing research, especially if you're doing cancer research. So that is a great advantage, that concept of everything is around you, and you're not lacking any technology, brain, or it's such a wonderful institution. And Baylor is one of the major institutions in Texas Medical Center, and so Baylor is somewhere here, and my institution is my building is in my office in the same building, and the lab is also in the same building. So the advantage of Baylor Molecular and Human Genetics is the Baylor Human Genetics department is #1 department in the US medical schools in the NIH, National Institute of Health funding. So it's a very top department, a lot of major findings, especially in the genetic diseases. We have made a lot of major findings from genetics department, Baylor Genetics department. So it's one of the most known genetics departments in the world. And so I'll come to my subject. So mitochondria. Do you have any disturbances in the slide? Are you getting this message? Okay. Okay. So mitochondria, I think you all know mitochondria, right? If you study cells. So what's the major fact of mitochondria? Mitochondria is the powerhouse of the cell, right? Where you get ATP, where you get the energy. So all the energy is coming from mitochondria. Almost 90% of the cellular energy is produced in the mitochondria. And the most interesting factor with the mitochondria is mitochondria has their own DNA. Okay, so you know that in the cell, most of the DNA is, when we talk about DNA, we talk about the cellular DNA, the nuclear DNA. But mitochondria is an organelle, that's the only organelle. They have their own DNA, a circular DNA. Okay, and so I was very excited when I was a postdoc about, and another important factor of mitochondrial DNA is that it is always a maternally inherited DNA. That means you get your mitochondrial DNA from your mother, not from your father, because the mitochondrial DNA, mitochondria of father, the sperm is located in the tail. By the time you get fertilized, it's shut off. So you get the mitochondrial DNA from your mother. But also the same thing, if you have any mitochondrial DNA mutations, that also you inherit from your mother. Okay, so it's very important. It's a maternally inherited DNA. And when I was a postdoc, I was very excited because I always used to think why mitochondria it's a semi-independent organelle because they have their own DNA, they can make their own proteins, and they're sitting around the nucleus all the time. If you look at, I can show you some pictures. The mitochondria is sitting around the nucleus. Why it is like that? So it's just like a bacteria sitting inside a cell, because they have their own DNA, they can function. So I was very excited to know why they are doing. So I was very fortunate to land up in a group, one of the world leading mitochondrial geneticist, Dr. Wong. I started working with her on the mitochondria. So see, when you talk about mitochondria, the most important thing is oxidose, that is oxidative phosphorylation. So what is oxidative phosphorylation? Because when you get the glucose, the glucose is a major source of energy when we talk, right? So when it comes to the mitochondria, it make ATP using the oxidation. That's the electron transport. So what happened here? They use the electron transport chain. It's in your mitochondria. So if you look at your mitochondria, I can show you. So the major parts I have focused, that is the mitochondrial DNA. They have their own DNA. I can't see my point. Let me see. Okay. So they have their own DNA. They have a TCA cycle. What is the other name of TCA cycle? Anybody know? Citric acid cycle, other name? Krebs cycle, right? So they have the citric acid cycle. Then they have this machinery that is called the electron transport chain. That's called the ETC. So what happened is ETC is a group of complexes. Complex 1, complex 2, complex 3, complex 4, like that. And each complex is a bundle of several proteins. For example, complex 1 itself is almost 40 proteins, 43 proteins, like that. Okay, so when you say complex 1, it doesn't mean that it's a single protein, it's a group of proteins. Okay, so like that, complex 2 is basically it's a small one, it's 4 proteins, and complex 3, complex 4, like that. So if you look at this slide, you can see the NADH and the FIT is coming from the TCA cycle, or the Krebs cycle is going to the complex 1 or complex 2, then they go for a series of electron transportation, then finally the ATP is produced. Okay? Can you see my mouse? So the advantage is, so another important thing with the mitochondrial ETC is that the proteins are a combination of nuclear DNA and mitochondrial DNA. Okay, if you look at here, complex 1, for example, it's around 36 proteins are coming from nucleus, and around 7 proteins are coming from mitochondria. And it's like all other complexes, as you can see in the slide. But the only issue or the only change is with the complex 2. If you look at complex 2, all the proteins are coming from nucleus, right? So because all other complexes are a combination of nuclear proteins and mitochondrial proteins. So that is why whenever we do mitochondrial assays or mitochondrial function studies, we use complex 2 as a control. Because if there is a problem with the mitochondrial DNA, that should not directly affect the complex 2, because complex 2, all the 4 proteins are coming from nucleus. Okay, so don't get worried. This is just, you wanted to at least to give you a brief of what is electron transport chain, because when I'll be talking a lot about oxidative phosphorylation in my talk, so you need to understand what is happening. So this is basically the generator of the cell. It makes the ATP, and that generator, that the ATP generation is happening from TCA cycle. It goes to one of these complexes, then it moves by complex 1, complex 2, complex 3, like that, and finally the ATP is produced. ATP is your energy. Okay, so when I talk about OXPHOS or electron transport chain or ETC, you need to understand that inside the mitochondria, this is a place where the energy is produced. Okay, so the mitochondria is something like this. You can see the nucleus, and the mitochondria is just surrounding the nucleus, and each cell has several mitochondria. It's not like, because you know that there you only have one nucleus, but you have several mitochondria around it. And that is why the mitochondria has an advantage. They can fuse together and make a long mitochondria. They can split apart and then give small fractions of mitochondria. And each mitochondria is an individual unit, because they all have their own. Then each mitochondria can have different mitochondrial DNA, not just one mitochondrial DNA. Okay, are you following me, or I'm making it more complicated? It's okay, right? So you have the cell. Inside the cell, you have nucleus, and there are a lot of other things, but I'm talking only about mitochondria. So you have the cell, you have the nucleus, you have the mitochondria, and mitochondria is the one generating the ATP or the energy, and the ATP is generated in electron transport chain, where the ETC, where the electron is, the transfer of electron is the major way it is producing the energy. And cancer mitochondria is very important, because mitochondrial altered function is one of the common consistent phenotype in cancer for a long time. Everybody know that. Most of the cancer has, because sometimes people say cancer is a metabolic disease, because cancer has, the altered metabolic function. And another important fact is mitochondrial DNA is susceptible for mutations. The major reason is, believe me, okay, because I know you might not have studied that. In nuclear DNA, you have histones, okay? So there's a big proteins called histones, and the nuclear DNA is, around to these histone proteins. That's why that. Have you ever seen the picture of chromosomes? Like, it's, shrink to chromosome, right? It's rounded. So usually the nuclear DNA is very much protected from reactive oxygen species, because it is a histone protection is there, but mitochondrial DNA don't have that protection. And another thing is, in reactive oxygen species, right? If you don't know, it's the active oxygen. It's a fire. Okay, so it's most of the reactive oxygen species is produced inside mitochondria. So the mitochondria is always exposed to ROS or reactive oxygen species, and also they don't have the histone protection. So that is why mitochondrial DNA is more susceptible for mutations. And mitochondrial retrograde regulation means there is always a communication between mitochondria and nucleus. Okay, so it's just like you give me that, and I will give you, like, it's a barter system. It's always a communication happening between mitochondria and nucleus. That's why I call mitochondrial retrograde regulation. So mitochondria can regulate nucleus. Okay. So this is the major important point you need to understand. If you don't understand this factor, you will not understand my talk. Okay, so that is called Warburg effect in cancer. Okay, so this is the most famous theory in cancer metabolism. That is in 1920s. Otto Warburg is a German scientist. He found that even in the presence of oxygen, because the ETC, the oxphos only happen in the presence of oxygen. Okay. So he found that even in the presence of oxygen, instead of oxphos, cancer cell demonstrate a distinctive phenotype. That's they use the metabolism they take a lot of glucose, but they don't use the oxidative phosphorylation. They don't use the mitochondria, but they go for a fermentation process and make lactose, and that process is called glycolysis or the aerobic glycolysis. I will explain that. If you look at this figure, pointer sometimes missing. Okay. So if you look at this figure, you can see that the glucose is coming, then glucose become pyruvate, and in the normal cell, the left side is a normal cell, and most of the pyruvate is going into the mitochondria in the presence of oxygen, and they make ATP there. Can you see that? The green color one? I'm worried about my pointer. Can you see my pointer? Sorry, I'm very much used to the Mac, so that's why. Sini, can you help me? Pointer on the. Oh, okay.
Pointer options.
Laser.
Laser.
Okay. Can you see me now? The pointer. So this is Warburg effect. It's in the normal cell. When the glucose comes, it becomes pyruvate, and pyruvate will go to the mitochondria, and they make most of the ATP in the mitochondria in the presence of oxygen. And whenever there is no oxygen, oxphos will not work. The mitochondria cannot function because there's electron transport. It needs oxygen. At that stage, they go for glycolysis, then they make lactate. That's what happened in your normal cell, in our normal cell. Like most of the glucose will go to mitochondria, they make oxphos. So what they observed is that in the cancer cell or a highly proliferative cell, even in the presence of oxygen, you got it? That's the most important factor. Even in the presence of oxygen, they are not going to the mitochondria, but they are making more lactate. Okay, so that is called aerobic glycolysis. That's even in the presence of oxygen, aerobic situation, even in the presence of that, they make a lot of glycolysis. But the cancer is taking a lot of glucose. But they don't want to go to mitochondria, they make the glycolysis. So that is called Warburg effect. Okay, so what happened with the Warburg effect is so the glycolysis has an advantage. They make a lot of biomass because it's a very quick process. You see here glucose, pyruvate, glycolysis. So the process is very, very, very active. You don't have to go through all this process. Okay, and this process you always see in the clinic because you see that PET scan. What you see, they are trying to find out how much the glucose uptake, right? Label the glucose uptake is happening in the PET scan because cancer cell is really taking a lot of glucose because for the energy. But there is a problem. Problem is there is a concern. Oxidative phosphorylation can make 36 ATP from one glucose. But glycolysis can, in the cytoplasm, can make only 2 ATPs. You got my point? So one glucose can make 36 ATPs if you go for oxidative phosphorylation. But one glucose make only 2 ATP if you go for glycolysis. So it's a very inefficient process, right? If you have one rupee, if you can buy 10 chocolate, and if you can buy 2 chocolate, it's a difference, right? So it's a very inefficient way of spending money, right? So but because Warburg's theory, for a long time, because it has happened in 1920s, a long time people thought mitochondria is dysfunctional in cancer. That was a concept for a very long time. So people thought, okay, if it's a mitochondria, if it's a cancer, mitochondria is not functioning, everything is glycolysis. That was the concept for a long time. But the question is, why the cell should do such an inefficient thing? One thing is they can make a lot of byproduct because it's a very active process. The reason is that what the theory happened or they proposed considering only the major factor glucose. But now we know that mitochondria is not just depending on glucose. They can use fatty acids, they can use glutamine, and all these can support the generation of ATP. Okay, so the whole mitochondrial concept from Warburg theory, thinking that all glucose is going to glycolysis. But that time they never considered that glucose is not the only source of mitochondria. Very recent years, in the recent decades, studies have shown that though glycolysis is one of the major factors in most of the cancer. If any cancer you go, most of the cancer they get a lot of glucose. If glycolysis is happening, mitochondria is still functional in many cancers. Okay, so one of our papers also, one of the first papers we showed that fat. I'll talk about that. Fatty acid is a major source of energy in some of the breast cancers. So that concept has changed, like in the sense glucose is the only source, and if glucose is not there, or the cancer is not functioning. Like in sense, if you just withdraw the glucose, mitochondria, the cancer cannot function. So that concept has changed now. So we did. So you understood this picture? I just want to make sure that you understood that the TCA cycle can be fit. The TCA cycle is making NADH and FADH2. This can be supported by glucose, fatty acids, and glutamine. And there is a lot of other things also, amino acids also can do that. So that is a major shift happened. The shift happened is that instead of thinking only about glucose, in the recent decades, people have started thinking about other energy sources. I just want to make sure that you understood this picture, because otherwise you will not understand the rest of the thing. Okay? All I want to say that the Warburg theory was focused only on this pathway, but we have this pathway, we have this pathway, and we have a few other pathways which can generate oxygen. So our lab, we did a strong collaboration with the computational biology team. We took this multi pathways for a mitochondrial ETC, and we made a model, a computational model, where we took two different major regulatory proteins. Don't get confused, okay? So what I want to say is, we took two proteins, HIF1. HIF1 means hypoxia-inducing factor. Hypoxia-inducing factor means hypoxia. You know hypoxia, right? Oxygen, hypoxia. That means there is no oxygen, right? So HIF1 is a factor which supports hypoxia, and it's a lack of oxygen. And AMPK is another factor which support oxidative phosphorus. So we took this. Don't get confused. What I want to say that HIF1 basically support glycolysis. AMPK basically support oxytocin. Okay, so we made a computational model using these two major factors, and we found that. So this is a complicated network. There are so many proteins involved. Don't even look at it. Okay, just want to show that there are so many factors. It's not that two proteins are involved. It's a big network is involved. But what I want to say that this is a mathematical model. Okay, don't worry, but this is called unapplied. All you want to understand here is that the green spots are the stable stage. Okay, you see these green spots are great stable stage, and the white spot is an unstable stage. So we plotted with AMPK which is supporting the oxphos, HIF1 which is supporting the hypoxia, and and this is called null plane, and this is the ratio. What I want to say is, in the normal cell, they only have two stable states. Two stable states. In the presence of oxygen, it is Oxford state. In the absence of oxygen, Warburg state or the glycolytic stage. But what we found is that in case of cancer, they can have three stable states. In the presence of absence of oxygen, presence of oxygen, oxidative stage, and there is a third stage. The third stage is called the hybrid stage. That means the cancer cell can have the stability with both oxphos and glycolysis. That is why we named it as a hybrid metabolic stage. You know, and we did, don't worry about this one. This one is basically one to say that we did a large number of patient samples and the gene expression data. We found that most of the tumor, they have oxidative stage, and they have some of them have a active glycolytic stage, but there is a bulk of tumor. You can, if you look at here, you can see that there is a bulk of tumor. The red color is uniformly distributed. They are called the Warburg stage. Okay, so I know that it's a complicated slide. Don't worry about it. So what I wanted to say that cancer cell has a lot of advantage of doing it. You know why? Because oxidative phosphorylation, they depend only on oxidative phosphorylation. Many cells cannot survive when it comes to hypoxia. That means then the lack of oxygen. If they depend only on glycolytic stage, they have a very inefficient system in presence of oxygen. So, but if they maintain a hybrid stage, they have the advantage. That means they are more plastic. They can be switched from here to there. In cancer, there is one of the metabolic stage of cancer, there is a problem because the core of the cancer will start growing very fast, very, very fast, they become bulky tumor. So there is no enough time to make blood vessels to all the cells. Okay, because it's a rapid proliferation. So what happened is many areas of cancer is hypoxic because they cannot get oxygen because there is not enough time to make blood vessels to that area. So in the cancer, many areas are oxygen dependent stages areas and many areas are hypoxic areas because the cancer cells are proliferating very fast and they become bulky. But the blood vessel is not generating so fast. So in a tumor, if you divide a tumor, you see a lot of areas you have hypoxia because the blood vessel has not reached there because they are growing very fast. So if the cancer cell has this plasticity or hybrid stage, they have a big advantage. They can survive in an oxygen stage, they can survive in a hypoxia stage. So that is why highly metastatic, highly progressive cells has this hybrid capacity. Okay. So all I wanted to say that in this session was mitochondria is very important. They make the energy, and the energy is basically made from electron transport chain. And the electron transport chain is consists of different complexes, complex 1, complex 2, like that. They involve proteins from nuclear DNA and mitochondrial DNA. And Warburg theory, Warburg suggested that cancer is mostly depending on glycolysis, not much on oxidative phosphorylation, thinking that it's only depending on glucose. But right now we know that cancer is not just depending on glucose, they also depending on fat, glutamine, and many other amino acids and other things. So that is why now people think more about in a way that many cancer cells have active glycolysis, but at the same time of active oxidative phosphorylation, and that is called hybrid situation. Okay, and the hybrid situation will give a more plasticity for the cancer cell for survival, because cancer cell is always looking for somehow to survive, right? They don't want to die, right? So they take all the advantage of metabolism. So I want to talk a little bit about the mitochondria and nuclear crosstalk. I told you, the mitochondria is just like bacteria sitting around the nucleus, and they always communicate with the nucleus. Because a lot of the the proteins produced from nucleus are going to mitochondria, sitting in the mitochondria. Not only the electron transport chain, many proteins can translocate to mitochondria. So there is an active crosstalk is happening there. So before I go to that, I just want to introduce our major model system, that is triple negative breast cancer. Okay? You know what is triple negative breast cancer? The breast cancer is actually classified into different. So in a general term, we consider they have breast cancer, they have liver cancer, they have lung cancer, whatever it is. But all these cancers are different heterogeneous. It's not the same. If you look at the breast cancer, if you talk to 10 breast cancer patients, some breast cancer patients are getting tamoxifen, some breast cancer patients are getting some. It's depending on what type of cancer they have. Mainly, the breast cancer is separated into 3 subtypes. A majority of the breast cancer is ER/PR positive breast cancer. That means estrogen receptor, progesterone receptor positive breast cancer. Estrogen and progesterone are female hormone, right? Mostly considered as a male also has it, but it's a female hormone. So estrogen and progesterone receptor, that means that hormones to work, you need receptor in the cell. So ER means estrogen receptor, PR means progesterone receptor. So majority of the tumors are ER/PR positive. That is why it mostly come in the females, like in breast cancer is mostly. Another group is called HER2. HER2 means HER2 is a oncogene. It's a cancer gene. What happened is we all have only one of these gene in our DNA. These patients have a duplication of the DNA of that area. So they have this oncogene is amplified 3 times, 4 times, 5 times. Okay, so that's HER2 amplification they call. So those genes are tumors are called HER2 amplified breast cancer. So around 15% of the breast cancer is HER2 amplified breast cancer. And the third category is called triple negative breast cancer. Can you tell what is triple negative when you look at this? That means they don't have ER, they don't have PR, they don't have HER2 amplification. That's why they are called triple negative breast cancer. ER and PR breast cancer, there are specific targets are already available because we already know the target. So there are tamoxifen and other, the other drugs which can affect on the estrogen pathway can be used for treat ER/PR breast cancer. HER2 amplified breast cancer was a very aggressive cancer for a long time, and it was a very dangerous breast cancer for a long time. But luckily, antibodies found against HER2. Okay, there are blocking antibodies found against HER2. So even though the protein is very much activated, now we can block that protein. So that is why now HER2 amplified protein is a very aggressive tumor, but now that can be blocked by specific antibodies. So HER2 is also now a little bit manageable. The problem is with triple negative because they don't have ER to target, they don't have PR to target, they don't have HER2 to target. The only possibility to treat them is using general chemotherapy, like doxorubicin, like that general chemotherapy, but they are not specific. So that is why currently the triple negative breast cancer is really required treatment because they don't have specific target. Recently, some inhibitors all came for triple negative, but that's not so much into the triple negative breast cancer. So we are more interested in the triple negative breast cancer because we want to see how metabolically triple negative breast cancer can be targeted. I have a lot of slides, but I don't want to cover all. But I just want to make sure that whatever I talk, at least you understand. Okay, I can stop at any time, but at least if you don't understand, let me know because I don't want to give a talk which you don't understand. Because I don't want to talk about too complicated things which you don't. Are you understanding what I'm saying? At least you know what are the 3 types of breast cancer? Okay. You know why I'm focusing on triple negative breast cancer? You understand? Because they don't have any target, right? Okay. So if you go into molecular, genetically they have 5 to 6 types So I don't want to talk. The only thing what I want to say is sometimes I use the word basal subtype. That means it's triple negative breast cancer, genetically when you classify. So they also call basal. It's mostly enriched triple negative breast cancer. Okay, so don't worry about it. And another thing is, when you talk about breast cancer, you see many times people die of breast cancer, right? But they are not actually dying because they get a tumor in the breast cancer. Because breast is an external organ, they can easily survive even without that. How they die? Because those breast cancer cells go to another organ, which is more important. And I'm not saying it's important. What I'm saying is vital organ like lung, bone, brain, right? And because these cells will escape. If you look at this picture, the primary tumor in the breast cancer can escape from the breast. They can go, this is called CTC, means circulating tumor cell. In the blood cell, your tumor cells will be circulating in the blood cells. And they go to maybe bone, they go to lung, they go to brain. So then you get an issue with the lung, you get an issue with your brain, you get an issue with your bone. That is the reason most of the breast cancer patients, almost 100% of the breast cancer deaths are occurring because of the metastasis. Because if you get a primary tumor in the breast, you can remove the breast cancer, the tumor. Okay, sometimes they can completely remove the breast. Now they can regenerate it, it's okay. But the problem is when it goes to the bone, when it goes to the brain, when it goes to the bone, lung, then you have real physical issues. So in breast cancer, always the concept is somehow block the metastasis. Okay, keep the cells in the breast not going to another organ. If you look at this graph, I just want to say, don't worry about this graph much. I just want to say that in every stage of the breast cancer, most of the death or metastasis death is happening in the triple negative breast cancer or the basal subtype of breast cancer. That is why I told one of the reasons is they are very aggressive tumors. Another important factor is they don't have specific targets. They are generally treated with chemotherapy. Okay, so this picture is complicated. All I want to say that what they did is they removed the mitochondrial DNA from the cell and they implanted the tumor into the mouse and they collected the tumor from the primary tumor, they collected it from the circulating blood cells and they found that they collected the tumor from the metastatic areas like lung or bone or something. Then they just looked at what happened to those cells. So basically they removed the mitochondrial DNA, but when they analyze the different cells, the row zero cell means that the cells without mitochondrial DNA, that's called row zero cells, they don't have the mitochondrial DNA. So what happened is, in the primary tumor, they did not get much mitochondrial DNA, but if you look at this metastatic cell, most of the metastatic cells regained the mitochondrial DNA. They took the mitochondrial DNA from the other cells. That suggests that the cells which undergo metastasis really need a oxphos. They really much prefer oxidative phosphorylation. You remember I talked about oxidative phosphorylation. So that means the cell which go to the mitochondria, the metastasis or going to the other tissues, they require the oxphos. They require the mitochondrial function. So all I wanted to say that mitochondria is very important during metastasis. Because the primary tumor, if you look at this red bar, the primary tumor didn't take much of the mitochondrial DNA. But if you look at this, this bar, I don't know if it's yellow color. Oh, okay. So this bar, if you look at it, you can see that most of the metastatic cells, they took a lot of mitochondrial DNA. That means that to get it metastasis, they need a mitochondrial function. Just to highlight the fact that mitochondria is very important for metastasis. So my lab is very interested in this factor, like mitochondrial, nuclear, and crosstalk, how it is happening. There are a lot of factors is happening. So basically, they both communicate each other and they make the tumor. So that's what one of the major interests in my lab. So what my lab is doing is whether mitochondria directly communicating its nucleus, mitochondrial proteins. See, these proteins can go to the nucleus and change the nucleus, or mitochondria is changing another protein, and that protein is affecting nucleus. or same way, nucleus is making protein which affecting mitochondria. So we are mostly working on this model, that is mitochondria and nuclear crosstalk. How it is happening, what is its function, what is its important in cancer. So that is one of our major area of research interest. So we use a very typical system for finding an exploratory. I know that it's a little bit complicated system, so I just made a little bit simplified form for you to understand. I hope you understand. What happened is, if you want to compare these four drivers, okay, you just want to know who's the best driver. Okay, so what is option? They all are driving their own cars. But the problem is, maybe you found this person as the best driver, but there is another problem. This person is driving a Ferrari. Okay, this person is driving another car. So how do you know this he is the efficient driver? Because his car is a different car. So when you focus on the driver, what is the best option? What's the best option? They all drive the same car. So now if this person wins, we know that it is his efficiency, not because of the car. Did you understand what I'm saying? Okay? You need to say yes if you understand. You need to say no if you don't understand. Yes or no? Yes. Okay, so what I wanted to say, I'm not telling you you need to find the driver. I'm telling you what I'm trying to say is this is the model we take. Okay? So what we are trying to do is we want to analyze all the system in a same nuclear background so that we can nullify the nuclear factors. Okay? So I'll show you what we will do. We take cells which is where we deplete the mitochondrial DNA. That's I already told. These cells are called row 0 cells. They have mitochondrial DNA depleted cells. So this is, for example, it's a cancer cell. We depleted the mitochondria. Here it's a benign cell or normal cell. We depleted the mitochondria. Then we put mitochondria from benign cell, moderately metastatic cell, highly metastatic cells to this one. Okay. So now we have a system. Different mitochondria, but they all have the same nuclear background. You remember the car what I was talking about. Now all the drivers are sitting in the same car. So that means this cell has the same nucleus, but they have a mitochondria from a normal cell. This cell has the same nucleus, but they have a mitochondria from a moderately metastatic cell. These are the same nucleus, but they have a highly metastatic cell. Similar situation here. They all have a normal cell nuclear background, but they have mitochondria from normal cell, moderate cell, and highly metastatic cell. Now we compare those cells. So now what is the advantage? If you find a difference, that difference is mostly coming from the mitochondrial function because they all have the same nucleus. You got my point? So because our objective is to find out what mitochondria is doing in cancer. So that is why what we did, we made them all the same nucleus. Nucleus is because if they have a mutation, the nucleus has a mutation, all the cell has the mutation. Okay, so we don't have to worry about that one, but we change the mitochondria. So now if we find a difference, not all, but at least most of the changes are happening because we change the mitochondria. Okay, I can show you some. So what we do is, we do this type of cyclid cells. This is called cyclid cell. That means the cytoplasmic fusion. It's not just mitochondria. There is also some cytoplasm come here, but we more focus on mitochondria. So you see that here it's a normal cell. They have a different mitochondria. Cancer is a different mitochondria. Then we do all the omics. Omics means multi-omics, like genomics, proteomics, metabolics, all these large scale analysis. Then we find pathways, which is more important, which is changed because of the mitochondria. Then we do a lot of functional studies in the animals and protein or whatever it is. Then finally we go to the clinic, patient data. This is a pattern I use in our lab. Okay, I can show you one example. Here, this is one paper we published in 2016. So what we did is we took a moderately metastatic cell, triple negative cell. We took the row zero cell, you know that row zero cell, the mitochondrial DNA is depleted, and we transplanted mitochondria from this benign cell, moderate cell, that is someone cell, and highly metastatic cell. What is on the side is, this is called soft agar colony formation assay. Are you familiar with the soft agar colony formation assay? That means, I know that you're genius, you may not be. But what is that? If you put the cells on a soft agar, if you grow the colonies, only the cells has the capacity to grow as anchored independent cell, because usually the cell lines are flat, they grow on the plastic. So if they can grow on the soft agar, that means they have a more independent growth capacity. I can say one, you can find here. This is the benign cell, right? I told this is the normal cell. You see, there is no colony here. Is it clear on the slide? See, you can see no colonies here. That means they cannot, that the normal cell cannot grow as an independent colony in a soft agar. But if you look at the moderate cell, you see they can form several colonies. And if you look at the aggressive cell, they can form a lot of colonies. That means they have the independent growth capacity. That is what exactly happening with the cancer cell. If you inject a normal cell, they cannot go and grow somewhere else, but a cancer cell can go and grow because they have independence. Okay, so that is what it is happening. So what we did is, we transplanted mitochondria from these cells. These are the mitochondrial chronates to this cell. That means the moderate cell. Then we looked what happened to the cancer. What happened to the cells, hybrids? We were very surprised to see that even though they all have the same nuclear background, you can see they all have the same nuclear background, the benign mitochondria completely shut down the colony formation capacity. And the aggressive metastatic cancers mitochondria really enhance the, because this is the control. If you look at this one, the mitochondria from a cancer cell really, 4 times they increase the capacity. But the benign cell completely shut down the mitocolony formation potential. So what it suggests that mitochondria has a controlling capacity for the cancer cell's properties. You agree? Because what we did, we took the same nuclear background, we changed the mitochondria. A cancer cell lost its capacity. Another cancer cell, the moderate cell increased the capacity. That means whenever she changed the mitochondria, according to the mitochondria, the tumor property has really changed. So that means mitochondria is really doing something to the cell that really controls its capacity. This video is not working, so I think I cannot show that. But what I wanted to say that this is another experiment called wound healing assay. So that means we make the scratches in the plate, in the cell, and we see how the the cell migrate to another, how the wound is healing. So that is an indirect way of measurement of the metastatic property of the cells. So I cannot show that video, but it's okay. All I want to say that this is the wound distance. If you look at this graph, this is the wound distance. All these cancer, the red one, the red color is the cancer, and the green color is the normal cell mitochondria. If you can see that by 24 hours, the wound is completely closed. By the if you look at the red graphs, they're completely closed the wound by 24 hours. But if you look at the green mitochondria ones, the wound is not closing. That means they are losing the cell. But don't worry about this experiment. I'll show you a better experiment. Probably this experiment you understand. See, what we did is, we take the mitochondria, so the hybrids, that means this is the nucleus. So means someone 59 is the nucleus, this is the mitochondria, this is the benign. The green color means always normal. Red is cancer. So you see here, this is a cancer nucleus, but a normal mitochondria. Here's a cancer nucleus with a cancer mitochondria. We took the same mouse. The mouse has different mammary gland. That means breast. Okay, both sides they have. Usually we take the fourth mammary gland for transplantation. Okay, so if you look at that, one side we transplanted this cell with the cancer nucleus, cancer mitochondria, left side. Other side we transplanted cancer mitochondria with the normal, so cancer nucleus with the normal mitochondria. They both have the luminescence. because we use the luminescence to identify it. It's very interesting. This is one of my best pictures. You see, every mouse, all mouse got one side tumor. You need to understand that they all have the same nucleus. They are same mouse. What we change is only mitochondria. And you can you see every mouse got left side tumor, but not even a single mouse got right side tumor. So that means, because we didn't have to do any statistics for this, it's 0 and all. We were so excited at this result. We published it. So that means mitochondria completely shut down the tumor property. They cannot even form the tumor. Even though they have a lot of DNA mutations, all these oncoproteins are active, everything is there, but they cannot function. They completely shut off. Okay? Another thing is, this is a new concept in cancer. It's not very new, but maybe quite new. That's called the cancer stem cells. Okay? So I told you the cancer is a bulk of cells. It's a bulk of cells, it's a bulky cells. But what happened is, you can see that many times you treat the patient, the tumor shrink, you ask them, oh, how is your patient? Yeah, his brain tumor is gone. But after some day, you see the tumor came back. Right? You may be surprised, it's already treated, it's gone. Where it came back? Okay? So that is the concept is called cancer stem cells. Stem cells, right? Because embryonic stem cells, how a body is formed, it's from a one cell, right? Single cell divide. A single cell is forming your eyes, single cell is forming your nerves, single cell is forming your skin, right? So that is why they are called stem cells. They have the plasticity, they have the flexibility to form any type of cell. If you take your skin cell and divide, it's always skin cell. Right? If you take your nerve cell and divide, it's always nerve cell. But if you take a stem cell, depending on the stimulation, it can form to any cell. Because you have to understand that one single embryo, we make the whole body. So that's why they are stem cells. So there is also a concept called cancer stem cells. That means they are more like a stem cell. So why it is important is, in a conventional therapy, what happened? Bulk of the tumor is gone. They respond to the cell, the treatment. But the stem cell will survive because they have the flexibility, they are plastic cells. And after some time, they regrow because they can make different cells again. Okay? So this is a problem because many times when you treat with the patient, after 10 years, the same tumor come back. You think, oh, why the same tumor came back? It's gone long back. They say that after 10 years it came back. 10 years there was no problem, but there is another tumor came back. Because these stem cells can sleep for a long time. They are very slowly divided. They can sleep for a long time, but they can wake up. That is why they are surviving conventional chemotherapy, because conventional chemotherapy mostly attacking on the rapidly dividing cells. These cells can escape that. They can slow their pitch and they can escape. They can sleep for a long time. Then they can come back after many years sometimes. So that is why people are now looking for targeted therapies, stem cell targets. So if you can kill the root, then they cannot grow. Otherwise, what happens? You are a tree, you are just cutting the tree on the top, but the root is still there, they grow it back again. Okay. So nowadays a lot of research in cancer is going on in the field of cancer stem cells. Okay. So this is a classical example. Here, this is a stem cell marker. That means in breast cancer, this is stem cell. That's a CD44 plus, CD24 minus cells. This is a marker. Don't worry, this 44 and CD44 and CD24 are two markers of the cell. Okay, a cell with the CD44 positivity and CD24 negativity is considered as breast cancer stem cells. Okay, so you put 200 cells this side, and you put 20,000 the opposite cell on the other side from the same tumor. you process sort it out and you inject it. You can see that they only form this side. Even this one is forming with 200 cells. This one is not forming even with 20,000 cells. That means that stem cells are so potential, even with very less cells, they can form tumor. Okay, so this was a paper published in 2003 from Michigan Group, and this was a big change in the cancer field, the cancer stem cell theory. I was a postdoc in Germany at that point of time. I immediately picked up on this finding. So then we did in the patients. Is it time to stop? Okay, I'll stop with this, only this concept. I will not go further. Okay. So what we did is we immediately did it in the patients. What happens? So we found that these stem cells are very important for the metastasis. Okay, so that is why even in the book they are saying that in 2005, Abraham, at that time my last name was Abraham. So Abraham was the first to publish the prognostic capability of the newly identified cancer stem cell market. So we published this paper in 2005 that these cells have the high capacity for metastasis. So now they change the cancer stem cells to tumor initiating cells because they can form and make new tumor in the metastatic stage. So it is due to timing, I'm just leaving all these. I just want to show that even in the cyclids, you can see in the cancer to cancer mitochondria, you can form even with 100 cell when you can form tumor, cancer to normal mitochondria, even with the 100,000 cells, that means 1 lakh cell, they cannot form tumor when this can form only 100 cells. That means mitochondria is really affecting its stem cell properties. Okay. And again, this is another example of the metastasis. This is another experimental model called metastatic tail injection. If you inject the cells to the tail of the mouse, they go all over the body, and the cells which has the metastatic potential, they can form lungs and everything. So if you look at this again, cancer nucleus with the cancer mitochondria, they form a lot of metastasis in the lung. But you see this mouse, the same cancer cell with the normal mitochondria, they don't form any metastasis. Okay. Let me stop this. I think we should stop at time. I just want to conclude. So let me stop this. So what I wanted to say is cancer stem cells maintain a hybrid metabolic status. I think you are clear that, right? I told you many times. They perform phosphorus and glycolysis, and hybrid cells status allow the flexibility to use several nutrients because they are fast use, and also they can produce energy, and also they can generate biomass. That is the advantage of hybrid stage. And I told you, hybrid models are excellent tool to study mitochondria and nuclear crosstalk. Okay, and I also published. I think I didn't show you much that mitochondrial energy reprogramming regulate onco pathways, cancer stemness, and metastasis. At least I showed you a few slides of that. Okay, so with that, I just want to acknowledge many people, especially my lab members. There is a lot of work in this, and I thank you very much for your attention, and I thank you very much. And I can see a beautiful building, and I was really surprised when I came here. I really surprised. Such a wonderful building, and I'm sure you guys will be one of the first batch students out of this beautiful college. And thank you very much. And you're always welcome. And if you want to know more about my lab's work, you can go to www.peripherallab.com. So we have some results and our publications are mentioned there. Okay, thank you. Again, I'm sorry. Actually, I didn't talk about everything, but I think at least one thing I said: mitochondria is important. You understood that? Then it's fine. Stop sharing.
Now this session is open for queries. You can raise if you have any queries. Online participants, please share your questions in the chat box.
I think everybody understood mitochondria is important. Okay. Thank you very much.
Hope you don't have any queries. So thank you, sir. Thanks for the insights into mitochondrial metabolic reprogramming in cancer. This session was proficient and fascinating. Sir, there is one question. So, a question from Sharon Richard. She would like to know how would you make a decision to use a cell line or go with an animal model?
First decision, money. Okay. So, see, the cell line is an in vitro model.